Thanks, Helen, and thanks for the wonderful words. I don't deserve them, but I'll take them. So I'm going to talk about, this is titled A Manual for Speaking to an Expert Audience. And the reason it's called a manual is because this is like how to fly a 747. All right, this is not, you got to feel your way around it, and we're going to cover 3 points, and at the end of the hour your mental state will be somewhat altered and it needs experiential. This is going to be bang, bang, bang. Keep the notes. Okay? The intent of this is that you will keep the notes and be able to refer back to them so that the next time you're giving a talk, you'll be able to look at them, think again about how you do it, and hopefully my words will come back to mean something for you. So let's just briefly introduce ourselves to somebody nearby. So introduce yourself and think, pair, share about 2 things you think are cardinal do's and don'ts about giving a talk. So just think for a moment. Things that you think about, you think, my top 2 would be, and then find somebody nearby, introduce yourself and share those things. Okay, let's come back, and what did you hear from your partner? Give me something that your partner said that sort of was an aha for you, or at least you were impressed by. Not your own thought, but one of your partner's thoughts. And I wish I had a whiteboard because I'd write them up, but we don't, so that's okay. Yeah. Yeah. Yes. Good time management. Great. We'll talk about that. Another one. Lisa. Manageable bites. Don't get too bogged down in the details. Great point. Great point. How about this sector over here? Anybody from something your partner said that resonated? Yeah. Great. So don't use too much jargon. And those are great segues into then the tip number 1, which is to know your audience. Right? So you have to know who you're speaking to. So these images, this is from a talk I gave in Japan last summer, and it was primarily to immunologists. So some of you may or may not know what an immunologist is interested in. But the talk that I gave, I specifically chose slides and knew what jargon I could use that they would understand, and what jargon I couldn't use because they wouldn't understand it. So knowing your audience and really thinking about it. Whenever I'm asked to give a talk somewhere, I'll say, is this for undergrads or graduate students? Is it for postdocs? Who will be in the room? What kind of people tend to come to your seminars? I always ask that because that way I can target my audience really well. I also need to know my goals. So you think, well, you're giving a talk. But that has nothing to do with goals. That's an action. What are the goals? Why am I giving that talk? And my goals might vary from trying to impress people so the Nobel Committee decides that I really should get the prize I so richly deserve. It might be trying to impress graduate students about what cool work we do because they may want to then come and be a postdoc with me. That would be great. It may be convincing undergraduates they should do research and not go to medical school the way their families tell them they should. Things like that. So you can have various agendas, but you need to be pretty intentional about knowing what are your goals. Ultimately, it should be not to tell them something, but to teach them something. And I'm going to come back to that point a lot. And if you leave here with nothing and remember nothing, other than don't tell your talk, teach your talk, I'll feel really happy, and I think you will have spent your hour and a quarter wisely. And the third is know your desktop. This is my desktop. You think, ooh, very messy. That's fine. What's not on my desktop is my email. And the number of people who open their computers and we see the most recent confidential emails is remarkable. Clean up, just make sure. Before I came over here, I deleted, closed everything except PowerPoint, so I wouldn't suddenly discover I'm doing grant reviews. That's why I'm flying to the East Coast to do an NIH study section. I don't want you all seeing my grant reviews. That would not be cool. All right. Second tip then, you know your story. you know what you're trying to do is to tell a story. Okay? Everything you do, you can frame it that way. And you can even, a great way to start a talk is, I want to tell you a story. I want to tell you a story. 7 words. If you do that, you instantly grab people in a way that you don't if you just launch into what we do is. I'm already tuned out with what we do is. But if you say, I want to tell you a story, you'll feel the whole room come into you, come toward you. And then you have to do it. And you have to make sure that it has an arc, it has a flow, and that the slides introduce it. And you have foreshadowing, which we'll talk more about. But create a story that you're going to give to them. So when you're doing that, of course, when you're choosing a book and a story to read, it's the title that gets you. So you need to have a title that's good. My titles tend to be long. I don't apologize for that. What I'm trying to do is capture as many people as possible because my goal is to educate people. And I don't want somebody not coming to my talk who should come to my talk and who would be better off if they did. Now that sounds incredibly arrogant, but why else am I giving a talk if not to educate people, right? So I want to make sure that people know what I'm going to talk about and are drawn into it. So choosing a title. So here's one that I gave. This one, I can't remember where this was last year sometime. Waltz or rave, how toxoplasma injects polymorphic protein kinases that determine the character of its dance with the host. Right? So there's a lot of jargon in there. That's okay because for the audience I'm trying to draw in, they will know that jargon. They'll know this is a parasite that we study. You're going to see it a lot today because I've chosen examples from it. And you might actually learn something about toxoplasma, which is a bonus for you. It's icing on this cake. I choose words like inject. So you could say, so this is a parasite that when it invades into a host cell, it takes over that host cell. It co-opts its functions. Actually, about 5 of you in the room right now are infected. And in your brain of each of you, of 5 or so of you in this room, you've got this parasite. And it's taken over the neurons that it has infected. How does it do that? It injects proteins to do that. So that word inject, it's a really active word. That was intentional in choosing the title. It's very dramatic. And then I'm using a metaphor here. Metaphors are good. Metaphors are a great way to teach people things. So this is a dance, and I'm trying to step away from the military metaphor. And I always say that the story I want to tell you today is about a dance. It's not about an arms race between parasite and human. It's about a dance because, in fact, you just sort of establish an equilibrium. So the intent is here, I'll draw in clinical people, I'll draw in biochemists who are interested in kinases, and I'll draw in people who are just generally in infection and the interaction between them. Why the backdrop? What's one of my goals? What did I say one of my goals might be? Impress them. Impress who? The Nobel Committee. They don't care that it's Stanford. Potential students and postdocs, right? So I'm selling. I'm saying, hey, this is actually, and it is, an extraordinary place, an environment to be in. All right, so we're going to tell a story, we're going to choose a title, then we're going to convey the challenge, the mystery, the coolness of it. So you want to really get excited about it and say, how does this parasite co-opt a human neuron? How does it do that? Right? I'm presenting a challenge and hopefully presenting it in a way that people say, yeah, that's actually a cool question. I use foreshadowing a lot. It's a technique that any writing class is going to teach you foreshadowing. We don't teach it anywhere near enough about giving a talk, and yet it's exactly the same tool for the same purpose. It maintains the attention. And in fact, in your brain, what it does for the neurons that aren't infected with toxoplasma, it helps create a structure in there. And you can say, oh, I need to remember that. That's not a point of whatever, another catalog point. He's telling me that for a reason. And I'll say, burn that into your memory because in a few slides, it's going to become relevant. And people do. They say, okay, and it becomes a little quest. When's he going to get back to that? You could even set it up as something like, well, they inject proteins, and how they do it is really cool, and you're going to be really surprised by the result. What did I just do? I foreshadowed it, right? So I'm creating a story and creating a little tension. All right, this is the point I made a few minutes ago. Teach your talk. Don't tell your talk. Don't tell people this is what we did. Because if you do that, you're going to go up there and say, and then we did this, and then we did that, and then we did this and we saw this, and then that's what we got. Thank you very much. Any questions? If you're teaching, you're engaging people. And if you put that into the front of your brain whenever you're giving a talk, that my job is to teach. They're here. It's precious time. Nobody has time to waste. Nobody's there because they didn't have anything better to do that day. They made an active decision to go. I'm going to teach them something because my audience, their goal is to learn. So I'm going to teach it. And I've seen this transform students' talks in a way that amazes me. They get up and they give their practice talk to the group, and we all critique them, and I say, you just told us what you did, but you didn't teach us anything. You love to teach. Teach us something. And they do it again, and they say, so what I'm trying to do is this. I'm trying to explain how to give a talk. Right? Suddenly they're engaged. They're engaging their audience in a way that teachers do and lecturers don't. So you want to be a teacher. What key things do you hope people will learn? Right? You're really going to emphasize that. A great way to do it is imagine the room is actually full of undergrads and you want them to leave having drunk your Kool-Aid and saying, this is just amazing stuff, I've really learned something. It's a great space to put your head in. At the same time, you want to think about what are the experts going to do? And what I tend to do is think about, well, those people in the room, some of them are virologists. So toxoplasma is a single-celled parasite. They don't care about toxoplasma in any way, shape, or form. They care about what they do, and they study viruses, which are tiny, non-living particles. What can I say that relates to that to them? And I will constantly relate back and say, when you think about how a virus does it, this is what they do, but toxoplasma has a different challenge. But then I'll say, maybe viruses do this too. So I'm getting into a space that they can relate to and that really means something to them. And then you just teach it all. You just teach. Pretend it's a class of freshmen that you're teaching to. Use the board. So it drives me bananas that modern architecture by modern architects who don't know how to teach keep putting screens in front of boards. A board should be beside the screen. Even if you're doing PowerPoint, which for a professional talk, 99.9% of the time or more, you're going to be using PowerPoint or using a projector. The board is typically in the room and use it at the same time. So if I had the board here before you came in, I would have drawn some models and produced an outline on the board. The reason is that the board is permanent. You fade out, you think about dinner. What am I going to have for dinner? It's gone. The PowerPoint's gone. You'll never get it back. The board is permanent. So you can build things on the board. The board slows you down. If you ever do any teaching, use the board. I taught a freshman seminar class an entire quarter, and I think I had 3 PowerPoint slides the whole quarter. I just used the board. It's a great way. It's a great tool. So use it, build on it. In other words, you want to communicate with people. You're just trying to engage them. You're trying to teach things to them. Don't lecture them, teach them. Don't speak at them, communicate with them. So focus on the learning, tip number 4. The way we learn is scaffolding. You have to have things to hang information on. If I gave you random facts, they wouldn't mean anything to you. If I told you that 30% of the world's human population is thought to be infected with toxoplasma, that would be a fact, but it wouldn't stick with you as much as saying that right now in this room there's half a dozen of you who are actively infected. That sticks. And then you say, and they're in neurons, and then what did I say? I said, I'm working on how they co-opt the function of those neurons. All of that's connected, and then how they take over you. That's what we're trying to understand. All right, so there's a scaffold there. You don't just present them with arbitrary random facts. You can use analogies. You want to repeat points. Obviously, whenever you're teaching, you can't just say something once. but you don't want to repeat it verbatim when you teach. You always want to go off script. The thing that people will teach you about writing as well as about teaching is if you're trying to say something complicated, stop yourself and just say, what I'm trying to say is. And if you do that when you're giving a lecture, it's really effective. So just think, what I'm trying to say is that these neurons that are infected, we don't know how that neuron can tolerate having a parasite inside the neuron. That's what I'm trying to say. So you're rephrasing it. Make it relevant, personally and or globally. Your work is undoubtedly interesting. None of you would do what you do if it wasn't interesting. Is it important? Those are not the same thing. You should be able to explain to any audience why your work, which is probably funded by tax dollars to some degree or by foundation dollars, is actually important. Because nobody pays you to do stuff for fun. They pay you to do it if it's because it's important. So you've got to be able to explain that. So if we think about this question that I might ask on a slide, what makes the 3 types of toxo so successful? So what I didn't tell you is that there are 3 major types of this parasite, different strains of the parasite, and they do really different things, but they're all incredibly successful in terms of being infection. Not only are 30% of humans infected, virtually anything warm-blooded, a high percentage of those animals are infected. The sea otters are infected, it's killing them. Cats are infected, dogs are infected. So this is a title I might give, and I'll say a unique combination of quantitative trait loci. Now that's incredibly jargony, and depending on my audience, I'd have to explain that, and I would. But then I would make the analogy, and I'd say, here are 3 individuals who are gifted in their respective sports, probably in all sports, curse them, but anyway, gifted individuals. Why? Because they have a unique combination of genes that came together to make just phenomenal athletes. They're superb. And so I would make this analogy that the 3 strains are like these 3 people who are just phenomenally good at it. And then I would go on to explain how they can't have sex with themselves yet. And so when they die, it's going to be a loss to the world of their phenomenal talent, whereas toxoplasma can have sex with itself and make more exactly Stephen Curry's or Abby Wambach's or Yao Ming's. Right? So that's a concept. I'm creating a structure for them, and I'm making an analogy that even for the most sophisticated, I'm joking about the Nobel thing, or you guys don't know me very well, but anyway, even if I seriously was trying to suck up for a Nobel Prize, I'd still show that slide because people like to understand and to be taught and to see analogies. All right, number 5, you're going to provide a plot line, a plot map. You can do that with a slide. You can also do that on the board that hopefully the architects didn't obscure with the big screen. You can just say, these are the points I'm going to cover, and then they can see where you're going. Everybody gets a little more relaxed. Partly they're ticking off saying, well, thank God he's at least point number 3. This probably won't be what I deduce, said what ticks you off the most is when people go way over time or condense everything at the end. So it's easy to see where you're headed. It allows you to remind them of what they've already heard so you can refer back. And I do that all the time. I say, so we've talked about this, and I've explained how toxoplasma gets into the neurons. What have I done? I've repeated it. I've given it structure. I've created a framework. Now I'm going to talk about this. Great excuse to repeat what you've already said, which is what every teacher has to do. And you can use highlighting. So here's outline of the story. This is one of my research. So I'm going back and forth between slides for today's talk and slides that I actually use in a talk to a specialized audience. This is one of the latter examples. So I say, here's the outline of the story. I'm going to introduction to the dancers. I keep my metaphor alive. The moves and their meanings. So tons of jargon in here. That's fine. And I'll even put that on the board so they can refer back, because even the most sophisticated expert audience largely will have no clue and will never remember what rope 18 was. That's random jargon for specific to my own lab, right? So this is a way they can refer back to it. And when I've had later on, I talk about rope 18 and it's on the board, They can say, oh yeah, I know that one. I remember that's, or was that the same? No, 16 is that one. Okay. Why do different dances exist? And then if I'm midway through the talk, I might have those grayed out and say, now I'm going to talk about rope 18. Great, we see this nice progression all the way through the talk. It's very reassuring to your audience. Tip 6, make slides work for you. One of my top 2 pet peeves is people who put 7 figures into one slide. It just drives me nuts. I get so irritated I stop listening. And even if I wasn't irritated, I wouldn't be able to see it and follow it. You want to have less. So less is more, okay, with those slides. You want to use a sans serif font. A serif is the little thing that is at the bottom of a letter. So a serif font would have a line across the bottom of that F, and that's to help you read a book so that your eye can scan and it creates a line underneath every line. That's why those fonts were created for books. This is not a book. Studies have shown, I'm told, I've never read the primary literature, but they say, and I choose to believe them, that that doesn't work well when you're doing slides because you're not trying to keep running across a line. So use a sans serif without a serif font. Make sure it's large enough to read from the back of the room. Okay? That should be always, you never want stuff that's so small that people can't read it. Same simple background for all. Now there's 2 camps on this. How many are in the white camp and how many are in the black camp? White, hands up, white background. Hands up, black background. Yeah, so that's about a typical ratio. And the black background people would argue passionately for why that's better. And I don't really care as long as it's consistent. The reason I like white is that if I put this on a black background, I couldn't print it out. And so much of what I do, I'm printing it out. So I like white, and that gives me more options, and I can cut and paste things in and the white disappears. So my recommendation is white. My strong recommendation is not a PowerPoint beautiful thing with dots disappearing on every slide in the background. It's really distracting. Makes them hard to read. Use color sparingly, and to make a consistent point, as in don't bother. So maybe you might have red for emphasis, but I once had a person in my lab who every word was a different color, and it was so distracting. And he had a color scheme that I never figured out, but it was different. Animate using appear mode. No sliding and no whoosh sounds, all that sort of thing. It just should be appear. It takes time otherwise for it to come in, and I personally find that distracting and not helpful. Minimize the text. So your bullets should be fairly short. You don't want paragraphs, but sometimes you actually want whole sentences. So sometimes my conclusions may be a whole sentence. And that's okay, so long as you actually read it. What's very hard for the brain to deal with is when the eyes and the ears are in opposition to each other. The eyes see something and the ears see something else, can't compute. So if you have a whole sentence, read it. It's okay to read it, bearing in mind too that for a technical audience, almost certainly many people's first language is not English. And sometimes I hear people talk about how to give a talk and they forget that half of that audience may not be able to instantly hear it and compute it, and they need the visual. Second reason is some people are visual learners. They need to see it rather than hear it. Some are auditory. You want to give both. But anytime you have full words, read that whole sentence if you need to have that in there. Avoid text on the very bottom of the slide. Often it's hard to see if you're at the back of the room. Depending on the configuration, it can be really a challenge. 5 bullets maximum. I know there's more than 5 bullets on this slide. This is not a technical talk. This is a manual on how to give a talk. They're written down. You're going to keep them. You can refer back to them. So I'm unapologetic about that, breaking that 5-bullet rule. But for a technical talk, I would not. I would break it into 2 slides if necessary, or try to condense it. So here's an example of a slide that I use. Injection of the rope tree contents is a very early event. So like I say, there's a little bonus for you today. This is toxoplasma infecting into a human cell. This is the human cell's nucleus. And when they do, they inject from these organelles that are called rope trees. Rope tree in Greek means club, as in caveman club. So these things inject during invasion. So here's the parasite invading in. What have I done on this slide? I've got the original citations. I'm not claiming this is my work. I've actually given 2 citations because different stages people have shown it with greater or lesser detail. I've labeled it intuitively. You didn't even need to be listening to understand what was there. And this actually this would be animated. I think I've lost the animation when I normally show it because I'll say, and Kamada and Tanabe were the first to show that that apparent emptying is actually injection of the proteins into the cell. So there's only one point I'm making on this slide, or one with a subpoint. Rope trees inject their proteins. That's it. It's very graphic, it's very visual, and it should be very intuitive. What else did you notice that I did there? It isn't a bullet and I need to add it. What did I do when I was telling you that, when I was teaching you that? What did you hear me do? Pointed to the slides, actually walked up to them, and that's okay too. Rather than standing way back here and with the laser, that's fine. As long as you can stand the glare from my head. What else did I do? Yeah. Not only did I define it, what did I do? Exactly. It is so helpful whenever you are teaching. Any class I do, every word I use, I give them the etymology if it's not a common word. Why? How do you learn? You learn with scaffold. What's the scaffold? Now I know rope tree means club. Oh, I think of club. When next time you hear, when I say later on in the talk, I talk about the rope trees, you're not thinking, what the hell are rope trees? You use that word before, I still don't know what they are. Oh, caveman club. Got it. That's the Greek word for club. It comes together. I actually even do the etymology of toxoplasma in every talk I give, because everybody thinks it must be toxic. It's not. I can tell you why, but if we have time, it's fun actually. Okay, tip 7. Every data slide is actually like a paper, meaning it needs a title, it needs an introduction, it needs a materials and methods, it needs a results, and it needs a discussion. Oops, I lost, and the approach taken, and a discussion. So you want, whenever you present a data slide, stop yourself from tending to do what we all want to do, which is to jump to here before you've gone through this. So you want them to actually know what it is you're presenting and why you're presenting it, what they're looking at rather than, and so you can see that toxoplasma is co-opting the host cell. What am I looking at? What did I do? I told you that was a human cell. I oriented you. I pointed you to the nucleus. I showed you the parasite invading. So I told you what you were looking at before I described it. So here's an example. And in this one, I would say, in fact, on the previous data slide, I would have said, so what we've found is that when toxoplasma infects, different strains do different things to the human cell. They change, it's really quite different what happens to that cell depending on the strain. So we were interested in looking at the basis of that because we hypothesized that a particular human protein might be affected. So what you're looking at here is infection with the 3 types of toxoplasma. Here you can see the phase image under the microscope, the human nuclei, the parasites for all 3. This is a stain for the nuclei, and you can see them nice and blue. And here we're looking at the green is the staining for the parasites, so you can see they're present and where they are. And the red is specifically looking at a protein called STAT3 in its phosphorylated form. What you can see, so what did I do? I introduced it, I gave them materials and methods, they know what they're looking at, and then I can say, what you can see, and you are already ahead of me on this, because I foreshadowed it, is that this one has no red. So what you can see is that that antibody, which is looking specifically at an activated protein that's really, really important for your immune response, isn't turned on by the type 2 strain. So what this tells us is that this may be the explanation for all the different gene responses that we see in a human cell. Okay? That was a paper. It was an introduction, it was materials and methods, the approach, the results, and the discussion. Every slide you want to do that. Tip number 8. Keep data slides clear and simple. There's only one thing that you've seen on each one of those slides, or I guess there's 2 panels on one of them making the same point, essentially. Cartoons and flowcharts are really wonderful. They just orient. Repeating them is great to have on slides. People get oriented better and better. You just want to make sure that they're conveying the real essence of the design. Include nothing you won't describe. It's really annoying to say, don't look at this. First thing what do people do? They look at that, right? Take it out. That's why God invented white squares in PowerPoint. You just plaster it over. It's easy to do. So nothing you won't describe and discuss. Label it intuitively. So if I put down there the antibody we used, 2G9.7 antibody, what does that mean? What I put down there was phosphostat-3. That's the antibody that sees that particular protein. Okay, that's what they need to see. Okay. So I'm labeling it intuitively. Walk through complicated slides slowly. It's a wonderful detensioner, and it's a wonderful way to engage your audience to say, I know this looks like a ton. Let me walk you through it slowly. It's a great reminder to you to explain it. And when you do that, you get buy-in. Rather than the audience thinking, what, is he serious? I'm supposed to look at that and get anything out of this? Say, relax. I'm going to walk you through it. And then you do. Use the rawest form of data that is intelligible. So this is something that I've picked up from some of my colleagues when they beat up on graduate students who present data as a ratio of a ratio, and they'll say, okay, so you can see that this ratio of a ratio is 3.7. They always say, I don't like ratios. What were the actual numbers? Because that, if it was 3,700 versus 1,000, that means something. If it was, or let's say the ratio is 4. If it was 4 versus 1, that's nonsense. They want to see the raw data. So you want to hit the sweet spot, presenting the rawest form that is actually intelligible, and then give the ratio as well. So people trust you. The thing that you will be judged on more than anything is your rigor. People will forgive. If you go over time, they'll be annoyed. They'll get over it, right? Some of the other things that annoy you, and there's too much data on the slide, they'll get over it. You talk not rigorously, they won't get over that. They will really hold that against you. It's the quality that you are judged on more than anything else. So if the ratio is 3.7 in one and 3.6 in another, you do not say, so they have the same ratio. That's 3.7 is not the same as 3.6. Just be super pedantic and rigorous as you describe things. You can circle or otherwise highlight key features. Use the animation to do that. It's great in a table to circle the thing they really need to look at. Very helpful. Rigor is paramount. And models are really valuable, and especially because you can repeat them and you can build them. Your story, you can add in new elements. You can start with something that's just a toxo and a human neuron or something, and then you can add in 4 slides later. So what I've shown you is that these rope trees inject the proteins. Now they see the rope trees injecting. And then I've shown you that one of those proteins activates a key transcription factor called STAT3. Build that into your slide. You're repeating and you're building and building, and the repetition really consolidates it. Include names of key contributors on the slide. So you guys are the contributors. I'm the representative. It's really important for me to give you the credit. Everybody knows that faculty, when they give talks, didn't actually do any of the work. We all know that, and that's fine. But that's not the same as saying, but who actually did do the work? So here's an example of a slide where I'm trying to present a model, and I've already shown you the data that this rope16 protein that comes from those club-shaped rope trees interacts with a human stat and mouse stat and activates it. And our hypothesis is that in a different strain, that protein is a little bit different, and it has evolved that way in order to interact with the stat from a different species, maybe chickens. Very simple, very graphic, very clear point that you're trying to make. And I put here the student who did this work, reference that on the slide. Tip number 9, make the oral delivery as exciting as the content. you can get into a mode that is just paralytic or comatosing, if that's a word, especially around lunchtime. If you're me, really especially around lunchtime. So you want to project your voice and enunciate. One of my best friends when I was in high school was deaf, and it helped me because very quickly I had to stop mumbling, and I actually had to enunciate and speak clearly, or she'd tune out. And it was actually a great thing to learn how to do. I also did my PhD in Scotland, and my Canadian to them American accent was unintelligible. And if I didn't speak really clearly and enunciate, they'd, what you saying, man? Anyway, modulate the pitch and the intensity. So I'm not modulating as much on the intensity. I'm being a little bit constantly intense, which will cause you to tune out a little bit. It'd be good if I just took it down a notch for a moment, right? So I get some variability. And then when I get back in intense mode, I'm making a difference. So try and modulate that. Do not memorize your whole talk, or unless you're a gifted actor. Memorize the beginning, which is when you're going to be most stressed, and then you'll get into it and you'll be cruising and you'll be teaching. You won't be worried about what was I going to say next. You'll be thinking, whatever it is, I'm teaching, and that'll be fine. So I always memorize the first few sentences of what I say. I want to tell you a story, and then I have the rest of that sentence or paragraph memorized. Never use list mode. What's list mode? It's a hockey stick. List mode is never use list mode, make eye contact, move around, pauses can be emphatic, pointers are for pointing, humor helps, da-da-da, da-da-da, da-da-da. That's list mode. And you'll hear people doing that, especially novices when they're giving an introduction. So, and by the way, the Brits especially hate this American thing of always starting sentences with so, but anyway, they'll get over it. So you might begin your talk. Toxoplasma is an important infection of humans. It also infects sea otters, and it can infect dogs. It causes a really serious disease. I'm just listing, and you're not listening as a result. So let's go back to the points I wanted to make there after never use list mode. You want to make eye contact. So you want to be using the whole room and making eye contact with individuals in that room. One of the really annoying habits is when somebody comes and there's the honcho sitting in the front row taking really careful notes. And so then we did this and we did this, and isn't this cool? And give me my Nobel Prize. And then everybody else is thinking, suck up, and like I'm, chopped liver. So make eye contact and take in the whole room. Move around. I have not been standing back here at the podium. This is an insecure position. I get here sometimes when I'm feeling insecure, but I try to remind myself to go out. Get out in front of it. Yeah. Yeah. So if necessary, you can have it nearby, and it's more important to be out there and occasionally have to look up at the slides to see what the next slide is than it is to be in presentation mode. So move around in different places. With this, it's a little hard, but this is relatively insecure standing back here, right? What about that? What's the power position there? See the difference between this and that? What do I own? I own this podium. It is my podium. Okay. So to the extent you're there, and as graduate students, you guys are low, really low on the totem pole, on the hierarchy, on the status thing. When you're at a conference, right? There ain't many undergrads there, so probably you're the bottom. That's okay. Own it, right? I own this, and I own my data, and I'm proud of my data. So move around, come out from behind the podium, or use that power grab. It's a really good one. When, what was the name of the senator? Arlen Specter, who came here to give a talk a few years ago before he passed away. Very important, very influential Republican senator from Pennsylvania, if I remember rightly. Very strong proponent of education and research, especially the NIH. And he was out, and at question and answer time, what he would do is he'd say, yeah, really. He'd get right in the person's face. It was it was fascinating to see how a professional politician completely disrespected it, but just he owned it in a way when the question time, really, right? It was fascinating to watch how a pro does it. Pauses can be emphatic. They can really make a point, okay? So use those pauses. Think about, where do I want to make a pause? 5, 6 of you are infected with this parasite in your brain right now. Right? It really is a very potent thing, and it changes the tone. So be prepared to use those, especially ones that are pregnant. Pointers are for pointing. They're not for waving. So, on this slide, you're seeing this one's getting a little tired anyways, but it's a very common mistake for people to do that. I go to the two-hand. Right? If I'm nervous, it's going to be wavy. I go to two-hand, and it's quite hard to wave with two hands. So that's where I can then say pointers are for pointing, not waving. So use the two hands if you want. Humor helps. Even a comic slide is a good thing to stick in there. I haven't done that recently. I don't know why, but I used to always, when Gary Larson was really hot, I always had a Gary Larson cartoon. Probably not worried about copyright or some stupid thing. Anyway, a humor is good. You have to be pretty judicious about it, but a little bit, like 1 or 2, is okay, and especially if it can be at your own expense. So what did I do? I didn't think about it beforehand, but I was thinking about it in the moment when somebody said, yeah, what you did was to go up to the board, and I said, yeah, if you can stand the glare from my head. I got a little chuckle. What does that do? It wakes you up. And I hadn't planned that. It's okay to throw a little humor and to be human in your humor. Polls can be useful. For example, which 2 things do you think are most important of the ones we've covered so far? And I've done this with, which protein do you think is most important for toxoplasma's ability to infect? But for you, I'll say, is it know your audience, teach your talk, use the board, show and build models, or own the stage? So this is a technical talk I'm, let's say, giving. And I would, halfway through, people are really beginning to lose it. Hands up, you can vote for 2. So have a look at the list. What am I doing? I'm engaging you, forcing you to engage the content here. You're going to be accountable. Look at the list and think about each one of those. Which 2 would you say are most important? And think for a moment. Okay, I'm going to do it by hand. You can vote for 2. Know your audience. Whoa. Okay, I think that's going to be one of the top ones. Teach your talk. Whoa, that's going to be another one of the top ones. Use the board. Okay, show and build models. And own the stage. Okay, good. So we had a smattering, apart from use the board, because there's no board, so I wasn't able to demonstrate how important it would be for you. Those 2 came out tops, right? You were engaged. And I could do that around all those jargony proteins I mentioned, and then say, in fact, and what I'm doing is foreshadowing, the answer is bang, and then I present them the answer. Hopefully it's not self-evident. You don't want to, when you're teaching and you ask questions, if I say, how many different base pairs are there in DNA? Anyone? Anyone? Right? So, like, am I going to be the suck up and say 2? Right? No, because it's too easy a question depending on the context. You want to make sure you ask a question that is worthy of people thinking about it. Yes, you had a question. No. All right. Okay. Oops, and I had a 6th one there. Sorry. Be excited. All right, tip number 10, be professional. You own the stage. You have to really feel like this is my space. You own the story. You don't apologize excessively for anything. It's a very common problem with graduate students. You are appropriately humble. Your ego is still in check. That will change. But just don't overdo it with apologies about, oh, this is, sorry, this isn't, not very clear, and I wish the data were better. Just, you have to posture. You have to be professional and say, I'm not going to apologize for this. Keep an eye on the time for our friend over here. And like most of you, how many have ever left a talk saying, that was too short? Boy, am I annoyed. And the opposite, many of us, right? So time is something you want to be paying attention to. And this is a great room because I can keep referring to that. And I'm self-monitoring here as a result. If you don't have a watch or clock in the back of the room, do something. You can have it up here. It's really hard, or a watch, but it's tricky. Hopefully there's a clock in the room. I once gave a talk, I kid you not, the clock was centrally wired and it accelerated 20 minutes in about 2 minutes. And I'm watching the clock and I think, okay, I'm good, and then give a few slides. And it was like I'd had a stroke. And anyway, 20 minutes, I kid you not. And they said, oh yeah, that clock is weird. It starts going and it stops. Okay. Tip number 11. It may be your 10th time, but it's there first. Regain your enthusiasm. Right. How many professional talks have I given? I haven't counted them. I would guess on my our science, 200 to 300. I could easily then get to, now I'm going to tell you about what we did, and there it is. You can't. You have to be animated about it no matter, and especially if you practice your talk, you can really fall into the trap of getting bored with it yourself. So that's why don't over practice, don't memorize it too much. So practice, 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 but then pretend it's your first time. Practicing, by the way, to your friends is far harder than giving a talk. If you can do that, you can do a talk, no problem, because you have to fake it, and you know that they know what you're going to say next because they've heard you talk about it so many times. Bring in some people when you do that, when you get your live group together, as close as possible to the real audience composition. So ask some people who haven't heard you talk before. At least then you'll feel like you can be teaching, because it's hard to pretend to teaching when Lisa's heard me talk about this at group meeting ad nauseum, right? She's gonna, it's really hard, and most of us aren't that good actors. We weren't trained for that. Memorize the talk's start and any key transitions. And for the remainder, just know what you want to say and then teach it to them. Don't tell them it, teach it. Tip 12, on the day, get in the groove. So when I showed up here in the nick of time to Helen's stress, it's because I was working with Robin Sugiro, because our course on Thursday, she's doing time management. We were talking about time management, which is kind of fun. But at any rate, I lost track of time. But you should arrive early, because that way you get in the space. And I feel I own the space. If I'm in the room when it's empty, it's my space that everybody else is coming into. If I walk into a full room, it's their space, and I get much more anxious, much more nervous in that. So I always say I want to get there early. In addition, it allows me to test the slides, make sure that the lighting's going to work, make sure that the screen, it fills the screen, the videos, get used to the sound system. Just feel like, yeah, this is my space and I'm in control of it and I'm not stressing about it. Learn the lighting system. So how do I turn down, you want to be figuring out, how do I dim the lights here if I need to dim the lights? You want to know that beforehand. You bring everything yourself. Never assume they're going to have anything. I carry with me 24/7. I have my pointer, my adapter, my VGA adapter, my power adapter. I have an extra pair of batteries for my pointer. Sometimes I carry an extra pointer because that's what I am. Carry my own water. Right? You don't want to be going dry and somebody has to run out and get you water. Bring a bottle of water. Why not? Put outline of key points or key points on the board as we talked, and bring backup flash drive. These days things are pretty constant. A couple of quick anecdotes. One time, couldn't get the thing to work. Some really helpful graduate student came up and he said, if you hit these, key coming, and destroyed my hard drive. I have still no idea what he hit, but completely fried my hard drive at the beginning of my talk because probably the projector wasn't plugged in. Another time, the AV person said, it's not connecting. I don't know why. And I said, I'm doing everything. And he says, well, it's your Mac. You need to reboot. Didn't work. rebooted again. And finally, from the back of the room was, oh, it's okay, I found that it's not plugged in. The projector wasn't plugged in. Okay. So I rebooted and I loaded up my talk and I got into my talk. And the fifth slide I thought, nope, it's the wrong talk. It was a talk I gave at that same institution 5 years prior. Get there early, figure this all out. You don't want to have to restart your talk. Okay. Give credit where credit is due. This is my wonderful group. You want to identify them at the start. So I show a picture at the start of the team. I include their faces. I include citations on data slides of anything published from you or others. It's really good form to always be doing that. You give the name and the journal of the year. That way they can look it up. And I often in a talk I say, oh, I want to look at that. So I write down, and I can find it easily. If it's just the person's name, it may not be so easy. Here's a great one, right? And this isn't about sucking up. This is about professionalism, respect. It's about saying, I know we don't work in a vacuum, and my colleagues and my competitors do really important work, and sometimes we find the same thing. I could show just our result with just my citation, very self-serving, not very professional. Or I could show it with my colleagues. Anybody who cares will know, and you just earn so many more points appropriately when you do that. Acknowledge collaborators. Right? Almost all of us work with collaborators. So here's an example of an introductory slide for the first chapter of the story that I'm telling. And these are the 2 postdocs and 2 graduate students who did that work, and then 2 competitors slash collaborators slash ex-postdoc slash whatever lab who've done similar work. And I'm just acknowledging that this, similar results were obtained by these folk, and we've interacted. It's great. Tip 14. And the total number is 16, by the way. So if you're looking, we're good on time. 14, answer questions deliberately. So if you get a question, try and answer it very specifically. A great technique is repeat the question. What it does is allows people who maybe somebody in the front row asked it and the people at the back didn't, allows them to hear the question. If it's being recorded, of course, otherwise the question makes no sense without the context. It also buys you time. It gives you a chance to think, just enough of your brain can be focused on what is it they're really asking, that as you repeat it, you can be doing that. And if they're going like this, as you say, so what you're asking is this, and they're going yes, then you're on spot. And if they're going this, then you didn't hear it right, or they didn't phrase it right. Never be defensive. If you get attacked, the most damage you can do is by getting defensive. The best thing is just roll with it. And this is a great phrase. Let's talk after, because I'd really like to understand your point. It's a really important one. And that shuts it off. And yet it says this person had a legitimate point they were trying to make, and I'm honoring that, and I'm not ducking it. Most of the audience will probably feel like that was a good call. We don't want to hear this fight. And it's not very often that people get aggressive, but it happens. And it's okay to not know. But if you don't know, follow up and just say, I don't know, but I'd like to get your name, and I'll be happy to look that up, because that's an interesting point. I'd love to follow up with you. You've all been there. Somebody says, so what does rope 16 do exactly? Well, that was 15 minutes of my talk. Where were you? Not an appropriate answer. Sorry, I wasn't very clear about that. Let me explain. Right? So I'm owning it. I'm taking some responsibility for that, and yet I'm not saying you're an idiot or you were asleep or whatever. Concisely answer the question asked, not the one you wish had been asked, which is something we all like to do. It's sort of, you learn it in grade school. You don't know the answer to the exam question, you just make up a question and answer it. Not very impressive, so try to really stick to it. 15, coming to the end of your talk, a summary slide. You could have words, or you could have graphics. So I like to have 4 quadrants. I figure 4 take-home points that I really want them to remember. This is my last chance. And it's a graphic, as you'll see in a moment. I include 1 bullet of half a dozen words or more if necessary for each one of those quadrants. That's my take-home. And I show it for my summary, and then I show the acknowledgements, and then I put it up again for questions and answers. 98% of talks you go to, what's the last slide what's up during question and answer? Sorry? Maybe some people will do that. That's better than nothing. No, it's a lost, every moment is a teaching moment, and you've lost the teaching moment. That doesn't teach them anything. It's usually the names, right? What do they learn from that? You want to be professional and acknowledge the contributions, but they're not actually learning. They don't honestly care what the names of those people were. It's wasted teaching opportunity. And then you're using it to burn in your message. So here's an example. I would have animated this and gone through each one of these. So what I've told you today is first that polymorphic injected ropes determine the character of the dance with the host. And they would understand what that is, have seen it. Then the second, then the third, then the fourth, all animated, click, click, click. Then I would show the acknowledgements, and then I would put this up without animation and say, so thank you very much, and I'd be happy to take questions. Now they can say, because they can't remember rope 16, rope 18. Rope 5, when you were talking about that, and they're pointing, right? I'm helping them frame the question. I'm reminding them of the key results and the key data. And finally, anticipate the next time you're going to give the talk. Every time I teach and every time I give a talk, afterwards I go back to the first slide and I put notes at the bottom. Next time, right? So next time, I said something here that I want to add in, and I've forgotten it now. Do you remember what it was? Okay. I will remember it when I go back to the slide. Add that in. Just say next time I want to do this. I didn't do this very well. This point I need a better analogy. I used to have Larry Bird as one of my basketball people. You don't even know who Larry Bird was. He's probably in an old folks home by now. So I substituted in Stephen Curry, right? And just something like that. Force feedback. You need to get the input. So I can ask my colleagues, how did that go? And they're all super nice people, and they'll say, really good, John, it was great. Feels good. What do I learn from that? Nada. Right? What would you change? That's what you want to ask whenever you really want feedback. And when people come into your practice talk and then you say, how did it go? And they say, it was great. Okay. Or even a real talk. How was I? Great. Okay, but what if I could change one thing, what would it be? And force them to tell you that. Lots of online resources. They're written down there for you. You can have a look at them. This is a great book. This lab management one has lots of things in a whole chapter on giving a talk. This one has, Burroughs Wellcome Fund has lots of books. One of them is on how to give a talk, Toastmasters tips, et cetera, et cetera. So that would have been, this is my summary. Know your audience and goals. Teach your story. Keep slides clear and simple, and use a graphic summary slide and leave it up at the end, which I'm now going to do. And thank you for your attention.